Hi, everyone, and welcome to today's Research America Alliance member webinar. And I think we have additional guests joining us. There's a lot of interest in this session and completely understandable because we have such a terrific speaker with us today. But first, let me thank you all who are Research America Alliance members for being part of our alliance. I think your membership is what lends us our strength and our ability to push for faster medical and public health progress. So thank you for that, for your ongoing partnership and for staying in close touch with us. And we try to do the same with you. So with that, today, I'm so pleased to introduce Dr. Diana Bianchi, who I'm sure many of you already know. She is the director of the Eunice Shriver National Institute of Child Health and Human Development at NIH. And as you know, this institute has been instrumental in the ongoing research base that has provided evidence on the COVID pandemic. And their work is important in so many other ways. So we are just thrilled to have Dr. Bianchi with us. As always, if you wouldn't mind typing questions into the Q&A box, or the chat function. We'll do our best to pose them when we get to the Q&A portion of today's session. And with that, Dr. Bianchi, let me turn to you.
Thank you so much, Ellie, for the kind introduction, and thank you to Research America and the Alliance for inviting me to speak today about the importance of research for keeping kids in school and pregnant women healthy during the COVID-19 pandemic. May I have the next slide, please? So I thought it would be important to start with the mission of our institute. The NICHD leads research and training to understand human development, improve reproductive health, enhance the lives of children and adolescents, and optimize abilities for all. And the reason why I start with this is many people don't realize that 30% of our portfolio is in reproductive health writ large, from development of the reproductive organs, to preconceptual health, to contraception, to pregnancy and postpartum. Next slide, please. So today I'm going to kind of split the talk and tell you about the research that NICHD has funded with the help of the Office of the Director to address the impact of the SARS-CoV-2 infection on children and women of reproductive age. So I'll be talking just briefly about multisystem inflammatory syndrome in children, otherwise known as MISC, about our Prevail Kids initiative as part of RADx RAD, what the trends have looked like following the summer of Delta, and what we're doing to get kids back to school. They're back in school, but to keep them in school, testing to stay in school. And then for women of reproductive age, I'm going to tell you the results of our initial study, the GRAVID study. And then we're going to talk a little bit about vaccination in pregnant and lactating women, and then about vaccines' potential effects on the menstrual cycle. So I'll be talking fast. Next slide, please. So initially, when the pandemic first started, most people thought, oh, kids, they don't get sick. It's really not a problem. And so there wasn't a whole lot of attention paid to both children and pregnant people back in February and March of 2020. It all changed in late April when reports began surfacing, mainly from Italy and the UK, that children were presenting to emergency rooms with some sort of syndrome that looked like another disease called Kawasaki disease, where there's inflammation of the heart, there's fever, there's swelling of the lips, there's a rash, there's abdominal pain. And there was great concern because these children were very sick. They needed to go to the intensive care unit. Some children died, but most children got better in about a week. Next slide, please. And so here in this slide from the CDC, you can see the dark blue line are the cases of MISC, and the dotted black lines are the cases of COVID-19. And you can see how MISC peaks slightly after, about 3 to 4 weeks after the peaks of COVID-19. And it's now known that it is a hyperinflammatory reaction that only some children get. It really only occurs in about 1 in 300,000 children. It affects males more than females and is more prevalent in Black and Latinx children. Next slide. 48 children have died just from MIS-C alone, and approximately 780 children, according to the statistics that we have died of COVID overall. Now, what helped us enormously were funds that were given to the Office of the Director to support Dr. Collins' NIH Rapid Acceleration of Diagnostics Program, or the RADx program. I'm going to tell you about 2 programs that were funded, one from the RADx RAD, which is all about developing novel technologies to diagnose SARS-CoV-2 infection, and one called RADx UP, which stands for Underserved Populations, in which we are going into communities and understanding what are the local needs to test children specifically in order to keep them healthy and keep them in school. Next slide. So the first program is the Predicting Viral-Associated Inflammatory Disease Severity in Children with Laboratory Diagnostics and Artificial Intelligence, known hereafter as PREVAIL Kids. And PREVAIL Kids has 3 goals. First of all, it's a translational project in which we're trying to understand at the time, back in the late summer of 2020, what is the spectrum of pediatric illness in SARS-CoV-2 infections. But importantly, because many people thought that these children had Kawasaki disease initially, how do you differentiate MIS-C from other conditions, such as high fevers, Kawasaki disease, or severe COVID-19, but without this hyperinflammatory delayed reaction? So the second goal was to develop diagnostics that would distinguish between the children who have MIS-C and the other children. And the third thing was develop diagnostics to be able to predict at the time of presentation, who is that rare child who is going to get MIS-C and is at risk for getting severe illness. And this is important because children would be totally well and within a matter of hours become critically ill and get admitted to the intensive care unit. So we really want to know who is at risk so we can watch them more closely. And this program uses a variety of different approaches, whether it's omics, whether it's immune profiling studies, which it uses artificial intelligence or other digital health platforms. Next slide, please. So we were able to fund 8 different teams. And as you can see here, they're from around the country, spanning from Michigan to San Diego, to Connecticut, to Texas. And these are all teams that have different multidisciplinary expertise, and they have access to diverse populations in over 75 sites across 30 United States, states in the United States. These groups also had previously acquired cohorts that could distinguish because they knew the outcomes. So they could distinguish between children who were known to have Kawasaki disease, for example, and they had large numbers of cohorts that already had previously acquired biomarkers and biorepositories. This is a study that includes both prospective and retrospective enrollments. Next slide. So I just wanted to show this to show you that in the prospective arm, with over 3,000 children hopefully to be enrolled, as of September 2021, we are ahead of target. So unfortunately, there are still children presenting with complications of COVID. And the bottom graph shows you that we have quite a number of children who've been enrolled with severe COVID and over 500 with MIS-C and 380 with Kawasaki disease. So there's a nice representation of all of these different diseases that we are trying to differentiate from each other. Next slide. And to date, we were aiming for 16,000, but we've actually enrolled more, over 21,000 children retrospectively to be able to probe their biospecimens, for example. And we are already, this study, I should have mentioned, was funded in December. So less than a year later, there are already results that are clinically very useful. So there already have been 4 gene expression signatures that predict MIS-C and differentiate it from severe COVID, for example, or from Kawasaki disease. One study is using, it's measuring proteins in saliva, which is very attractive because kids love to spit. And it has shown that certain cytokines or proteins in the saliva predict disease severity. And as shown here in a cover article from the Journal of Clinical Investigation, intravenous immunoglobulin is a highly effective treatment for MIS-C, and it works by reducing the number of activated neutrophils that are circulating and going to tissue and causing disease several weeks post-infection. And not only that, the investigators have already initiated discussions. So 4 of the 8 have initiated discussions with company partners or industry support to be able to bring some of these tests to point of care. Next slide. Not only that, these 8 investigators are working together beautifully, and they have collaborated to create a pediatric cohort that is now part of the Recover Initiative, which, as you may know, is an over $1 billion initiative that is working on the long-term effects of SARS-CoV-2 infection. So the Prevail Kids investigators are using their cohorts and their information to track these children long-term. Next slide, please. So I just wanted to point out that we can't take our foot off the pedal. What came as a shock this summer was what we call the summer of Delta. So if you look at the curve of children being infected and the number of cases added each week, the summer exceeded anything that had happened before, including the winter peak of 20 to 21. Currently, there are over 6.4 million children who've been infected, and children account for 16.6% of cases in the United States. So it's very, very concerning. Children are exposed to people who are not vaccinated, and until recently, children under age 12 could not be vaccinated. So they unfortunately were at great risk, and this is shown by this curve. Next slide, please. I should mention that weekly we do track data that comes from the CDC as well as the American Academy of Pediatrics. So we've been concerned from the very beginning of the pandemic, from March of 2020, when everyone was sent home from work and school. And because a large part of our portfolio involves child development and typically, as well as atypically developing children, what is going to be the effect of children being out of school, remote learning? Children are not necessarily getting the nutrition that they would normally get at school breakfast or lunch. They're not getting physical exercise. They're not having socialization. They're not getting special services. And so through the RADx Underserved Population Program, we have developed an initiative that NICHD leads, but it is a trans NIH initiative in which $58 million has been committed so far to provide evidence for the effectiveness, sustainability, and scalability of COVID-19 testing approaches and mitigation strategies in school settings. And as well, it's very important to generate information about social, behavioral, and ethical implications of implementation of COVID-19 testing within identified communities. And that varies in different places in the country. So the map, if you can read it above, is showing you the 16 sites across the 13 states. And importantly, we are working with schools that include public schools, charter schools, tribal schools, early education, and importantly, special education schools because children with intellectual disabilities have difficulty with mitigation strategies. So whether it's hand hygiene or masking, there are different considerations for those populations. And we've had to deal with a moving target, because when we were initially sketching out the study, vaccines were not available for any children. And then they became available for teenagers or high schoolers. And most recently, they've become available for children under age 12. We've also had to deal with the fact that children did come back to school in September, but there are a variety of criteria for sending children home or shutting down classrooms, etc. I'm going to show you in the next slide some preliminary results from the phase 1 study and some of the lessons that have been learned from this rapidly evolving but very productive study. And what has been shown so far is that direct communication with parents is far more effective than emails for increasing enrollment in testing. And strong relationships with school nurses and other medical consultants are vital for testing results and disseminating those results. It's really important to engage with the local community and develop trust. And trusted school champions are so important in recruiting staff and students for testing. I know that one of the studies that we supported actually engaged the teenagers who were given the assignment of determining whether they preferred nasal swabs or salivary testing. And surprisingly, the results conclusively showed that they preferred the nasal swabs because they could get it over much more quickly. And so we saw a video of some of the high schoolers, I believe it was in Missouri, telling us in St. Louis, telling us about what it was like to engage in that study. It's important to engage the students, the parents, the teachers, etc. And it was very important to frame COVID-19 testing as a school safety measure with other mitigation strategies, such as hand hygiene and masking. And when these mitigation strategies are in place, in-school transmission has been low. And that's why we believe that the default should be that children need to stay in school and we need to keep them in school. And so we can use testing, including households, to keep children in school. Next slide. So I'm going to pivot now to our obstetrical research and tell you a little bit about the GRAVID study, which is gestational research assessments for COVID-19. We support and have for many years support a network known as the Maternal Fetal Medicine Units Network. It consists of 12 centers with 39 individual affiliated hospital sites. This is a very important network. The results of their studies, 25% of their publications have been incorporated into professional society guidelines. So what they do, and they make recommendations about how care should be provided to high-risk pregnant women, it is definitely paid attention to by the professional societies. Well, we had these existing network sites, so we decided to do a medical records analysis of women who had given birth at one of these centers in the year before COVID compared to the year after COVID. And what has been finished so far is a nested subset of women, 1,200 women who were SARS-CoV-2 positive during their pregnancy. And they were divided into groups, whether they were mildly symptomatic, moderately symptomatic, or severely symptomatic. And the women who were mildly or moderately symptomatic basically were not at increased risk compared to non-pregnant people of the same age. But the women with severe symptoms were at higher risk for cesarean delivery, postpartum hemorrhage, hypertensive disorders, and preterm birth. Next slide, please. So we now know that pregnancy increases the risk of hospitalization, the need for mechanical ventilation, the need for ICU care. And to date, 218 pregnant people have died. And these were unnecessary deaths. I think that some of the information that came out early on was restricted because at the start of the pandemic, evidence for the utility, safety, and effectiveness of the available vaccines in pregnancy was really unknown. And yet our institute co-led a task force that was specified in the 21st Century Cures Act to do research on pregnant women and lactating women. And we made a series of recommendations. And so back in February of this year, we re-looked at those recommendations and realized that some of those lives might have been saved if we had been able to adhere to those guidelines. Next slide. So what we want to do is prevent deaths from the next pandemic. So it turns out now that via the common rule that pregnant people are no longer considered a vulnerable population. So it's not too early to begin preparing for the next epidemic or pandemic. And there were some initial pregnant rodent studies with the Moderna vaccine. So in the next pandemic, if there are animal studies or initial phase 1 safety data, and those are reassuring, there should be a plan for inclusion of pregnant people. We were fortunate that the CDC has maintained this VSafe registry, and they're able to pull out how many women have been vaccinated during their pregnancy. And that has provided a significant amount of information that has reassured us that there is no increase in complications in pregnant women who get vaccinated during pregnancy. There are also special compensation programs that could be used by pharma to be able to include pregnant people in these initial clinical studies. Next slide. And we also know now, what we didn't know then, some of the benefits of vaccination for pregnant and lactating people. And we know that the immune responses to mRNA COVID-19 vaccines are comparable between pregnant and lactating people and age-matched non-pregnant controls. Natural infection does generate higher antibody titers vaccines generate higher antibody titers than natural SARS-CoV-2 infection during pregnancy. And data from Israel suggested that mRNA vaccination during pregnancy reduces infection risk. We at NICHD have been funding quite a bit of research on how pregnant people, lactating people who receive the vaccine mount an immune response. And current research is looking at how long the protection lasts, but some very recently emerging research has suggested that breast milk is an amazing source of antibodies. And so I'm often asked, as a pediatrician myself, what can I do for my newborn infant or my 6-month-old infant who is not qualified to receive a vaccine? And the answer is simple. The mother should be vaccinated. And if she is breastfeeding, the baby is protected through antibodies that are crossing into the breast milk. Now, what we also know is that there are some differences between pregnant people and age-matched controls who are not pregnant, how they react after the first dose compared to the second dose. And after the first dose, they do not mount as robust an immune response. But after the second dose, they are equivalent to people who are not pregnant. So the bottom line is that pregnant people should be vaccinated if they are pregnant. Of course, it's best to get vaccinated before you're pregnant, but if you are pregnant, you should definitely get vaccinated. And if you're lactating, the same message. Next slide. Lastly, I just wanted to address the issue of vaccination and changes in the menstrual cycle. This is a topic that has generated interest from social media and traditional journalism. And I have to say that after a few interviews that we did, I have never gotten so many emails on a topic, as well as emails volunteering to participate in research from different women. We decided, because of questions on social media, and when we looked into it, there was a lack of evidence to say what exactly is happening. So we decided to award $1.6 million in one-year supplemental grants to 5 different institutions, to investigators who are already studying changes in the menstrual cycle unrelated to the vaccine, to explore how COVID-19 vaccination affected the menstrual cycle. And these new awards have the goal of providing evidence, whether this is true or not, or whether it's just anecdotal through social media. Are menstrual changes occurring as a result of vaccine? How long do these changes last? And what are the underlying mechanisms? Next slide. So, the reason why we decided to fund these studies is that it is biologically plausible. Menstruation itself is an inflammatory process with the recruitment of all kinds of immune-related cells, and these are important to break down and regenerate the endometrium, which is the lining of the uterus, every month. Also, the ACE2 receptor, which is the target of the spike protein of SARS-CoV-2 in a range of different tissues, is expressed in the uterus and may play a functional role. There are also functional roles for immune cells at the level of the ovaries and potential crosstalk of signaling, even at the neuroendocrine level. So it's an important scientific question that affects a significant proportion of the population. And we felt that it was very important to provide evidence, whether these changes occur or not. Whatever's going on seems to be temporary, but it would be much more reassuring to women of reproductive age if we could explain what is happening and how long it is expected to last. And this brings up the question that really harkens back to our work on Preglac and how various studies of therapeutics and vaccines affect women of reproductive age, and how important it is to consider the menstrual cycle, for example, as an important vital sign. And questions about disruption in the menstrual cycle really should be included in clinical research forms, as it is important information. So may I have the next slide, please? I've only had a limited amount of time to give you some highlights about these 2 populations. I'd like to direct you to our website if you're interested in a more complete picture of what we're funding. I suggest that you go to this website that is updated regularly. And with that, I'm happy to take some questions. And again, I'd like to thank Research America and the Alliance for inviting me to meet with you today.
I'm so glad. So do you have a couple of minutes to stay with us for questions,
Absolutely.
Dr. Bianchi? Terrific. That was a wonderful presentation. I have about a million questions, but I will limit myself. And I also want to invite Anna Platt to join us because she'll field audience questions. And maybe if we could grab 10 minutes of your time.
Absolutely.
Wonderful. More general question. Are there, I'm thinking about menstrual changes. Are there differences in the immune response to the vaccine between men and women generally?
So there are a lot of differences in the immune system between men and women generally. So and there's great interest as to why men die at a higher rate of SARS-CoV-2 than women. So the way that they initially react to the infection is different, and the possibility of long COVID is also potentially different as well.
So there are a lot of sex differences.
Oh, absolutely. Sex as a biological variable is very important.
Very important. You mentioned that for children that you've that research has produced evidence for 4 genes influencing that help predict severe disease. Is that defined as the kind of the multi-inflammatory syndrome itself?
No. So whether you get the multi-system inflammatory syndrome in children or not, really depends on your own immune system. Again, it's very rare. And your immune system is affected by your background, your sex, your ancestry, and that in combination with how you were exposed, when you were exposed, et cetera. But what these researchers are trying to do is they are trying to look at biofluids, whether it's plasma, and look for a signature that says, aha, this one has MIS-C, because that is still a point of contention when children come to the hospital. They're clearly sick. They clearly need to be in the ICU, but there isn't a definite diagnosis. So those 4 genes are helping you to determine if they have MIS-C or not.
I see. I was kind of, in a sense, I mean, hopeful in any of this is a terrible word to use, but if there were genetic markers for the prediction of severe disease, you would think that that might attend to all populations and health disparities and that kind of thing. But this is
so I think the confusing thing, I'm a geneticist by training, and when you think about genes, you think about DNA, you think about the DNA that you were born with, which doesn't change throughout your lifetime. And you are born, with risks for certain conditions, and you will be influenced by your exposures throughout your lifetime. But here we're talking about gene expression, and we're talking about RNA. And the RNA signals that are made in response to MIS-C. And that's what the researchers are looking for.
That makes sense, because that's so central to the vaccination process itself, is that RNA. And one more question. Do any of the major studies that you've mentioned, and there's no right answer here, it's just curiosity, do they extend globally? Is there any global involvement?
Yes, absolutely. So in Prevail Kids, there are a number, I kind of skipped over that, but there are a number of international collaborations with other cohorts in the United Kingdom, for example, in South America, et cetera. But we also at NICHD sponsor the Global Health Research Network, which I didn't have time to go into, but we have, again, a well-established network of 8 different sites in 7 low and middle income countries that are studying, the effects of SARS-CoV-2 infection on pregnant women in places like Guatemala, in India, in Pakistan, in several sites in Africa, for example. And we're, again, trying to see what are the effects in an area where there may not be vaccination yet. What have been the effects on their pregnancies?
Wow.
We're doing that by looking at the antibodies in their blood.
That network, the existence of that network created efficiencies in the COVID response. Very, very interesting. Okay, I'm going to zip it now. And I know that you have questions from the audience.
Yes, absolutely. Dr. Bianchi, one question that has come in. Recent studies have illustrated the negative mental health effects of the pandemic on children and teens in particular. And just last month, the American Academy of Pediatrics declared it a national emergency. Can you speak about that a little bit and what might be done moving forward to address that?
Yeah. So again, this is part of the concerns about not being in school. Children are in relative isolation. They're looking at a screen all day. We don't know the effects of the prolonged exposure to digital media on the developing brain. There's no question that many people, including children, but not exclusively children, have had serious mental health effects of the pandemic. We are funding some of that research, but we also are collaborating with our sister brother institute, the National Institute of Mental Health, which also supports quite a bit of pediatric research. I think it's important to note that we fund 18% of the child health research across NIH. Almost every institute has some aspect of child health research ongoing at the NIH, and that's why we formed the Trans NIH Pediatric Research Consortium, which allows all of the institutes to get together and talk about with a collective voice, what do we want to address in terms of improving child health?
Great. And I noticed a really interesting, but not that they're not all interesting, but an interesting audience question that came in a couple different forms. And that is the notion of using different vaccines for your booster or for your first and second vaccine. Is there any evidence around the specific impacts on pregnant women or dangers around that, or recommendations around mixing vaccines?
Yeah, so that's a great question, actually. I thought you were going to ask about children. So important to recognize that only Pfizer right now is approved for children. Moderna has been doing a study as well in children, but the results of those studies are not yet available. So if you are out there and thinking about whether or not your child between ages 5 and 11 should be vaccinated, it's going to be the Pfizer vaccine at the lower dose, so the 10 milligram dose. For pregnant people, I'm not aware of studies that are looking specifically at the mixing and matching issue. The NIAID is doing now, they're doing a study on pregnant women called the MOMI trial, where they're looking at specific immune responses to vaccination during pregnancy. But I'm not aware as to whether they're looking at the mixing and matching issue. I mean, I think that Tony Fauci, it's important to get vaccinated and take the vaccine that you're offered. It's less of an issue now than it was in February of 2021. But in general, they're recommending that you should get a booster with the original preparation that you got, unless it was J&J.
Yeah, because I know that the research that came out was actually preliminary about mixing and matching. It hadn't been peer reviewed at the time. It was very cool data, but
yeah.
And I think, could we have time for one more question? Do you have, I know there's several.
Yes. Yep. So another question that's come in, Dr. Bianchi, studies have shown that the pandemic has had disproportionately negative impacts on mothers of color in the United States. And from a health and healthcare perspective, what do you think are the best ways to address that inequity moving forward?
Yeah, so health disparities related to the pandemic, but health disparities in general, whether it's maternal morbidity and mortality, is a major focus of our institute. And this is where we really credit the RADx underserved population study and the community engagement study. The RADx UP study, the return to school, is not focused on mothers, but certainly mothers are affected by whether or not their children are at home with them all day. So that study is specifically focused on underserved populations, and we have quite a number of sites that are in communities of color. In terms of how women of color who happen to be pregnant are affected by our research, this is a part of a larger initiative called the Improve Initiative, which is also trans NIH, which is implementing a maternal health and pregnancy outcomes vision for everyone. And that initiative came after a meeting that we had with the Black Maternal Health Caucus in Congress, where they were urging us to do more. And we've been able to fund 2 years worth of supplemental studies. And the reason why it's relevant to the question is that this year the focus was on how have pregnancy experiences changed as a result of COVID-19, and also how has maternal mental health changed during the pandemic. So that research is ongoing. We don't have the results of those studies, but we are addressing that.
Well, Dr. Bianchi, this has been a really special meeting. I'm so glad that you were so generous with your time. I know you don't have much to spare, and thank you so much for being with us today.
It's my pleasure. And again, I want to thank you for inviting me. And I'm really speaking on behalf of just a fantastic team of people who are engaged all across the NIH, as well as our extramurally funded researchers. So thank
Well,
you.
terrific. Thank you again. I have just a bit of housekeeping that I'd like to run through. First of all, I hope that you'll join us next week for a special discussion with Bill Novelli. I think you all know him. He was a co-founder of Campaign for Tobacco-Free Kids, CEO of AARP, with CARE. I could go on and on about Bill. Porter Novelli and Matthew Myers, equally incredible. He was co-founder and is the president now of Campaign for Tobacco-Free Kids. They're going to come talk to us. Our board member and the executive director of the American Public Health Association, Dr. Georges Benjamin, is going to be having a discussion with them about how they managed to start this incredible social movement against kids getting involved in smoking and smoking generally. And I think it's just going to be a really informative discussion about advocacy, movements, and public health. Also, I wanted to mention, as you know, Thanksgiving is crazily just around the corner. And I hope you'll join us for Public Health Thank You Day, which we always hold the Monday before the holiday. I think you'll find in the chat, I'm reading from a script, so Anna, I can't see, but probably in the chat, there is a link to Public Health Thank You Day. Is that right?
Yes, that's in the chat, Ellie.
Great. And also registration for the Alliance member meeting next week. And I also hope that you'll keep your eye out this week, particularly for our weekly letter. Not only will this information be repeated, but we're going to have several really cool advocacy opportunities. As you all know, if FY22 appropriations is not completed on a timely basis, please say no, there's a year-long continuation of FY21 funding, then our nation stops, even though things like pandemics move. So we have some advocacy opportunities to try to push for completing FY22 appropriations before the December 2nd deadline. And there are other interesting information. And let me just close by saying that we are in our year-end annual giving mode. So I hope you'll consider making a donation for our work, and you'll find information about that in the weekly letter. So stay tuned for Thursday, and thank you for joining us. Take care.